Insect pollination represents an important step in the production of deciduous fruit. I and my colleague Mike Allsopp undertook in this project to work out the contribution of wild honeybees relative to managed honeybees in a typical deciduous fruit orchard. I'm Veldtman. I work for the South African National Biodiversity Institute, and I'm a research associate at the Department of Conservation, Ecology and Entomology, Stellenbosch University. Mike Allsopp works for the Plant Protection Research Institute, for the Agricultural Research Council, and is based in Stellenbosch. Pollination demand for deciduous crops will increase in the next few years, representing a demand that beekeepers will struggle to meet. Periods when multiple crops are flowering will be especially impacted, which will result in more expensive honeybee rentals. It is thus important to work out how many hives should really be stocked in an orchard. And this is our question in this project: how many colonies are really needed in an orchard if there is a wild pollination service available? So the ecosystem service of pollination represents a free service that the environment can deliver to the farmer, and it is simple to count the number of honeybees and differentiate them from non-honeybee pollinators. But to separate wild and managed honeybees is tricky because they look exactly the same, but they represent two different services. The one is provided by nature and the other one is provided by a beekeeper. So we propose to work out what is the contribution of wild pollination services to a typical orchard by closing and opening known managed hives and then counting the number of honeybees that are foraging in these orchards. Our method is to use a hive closure device, which importantly must not impact on the foraging behavior of the manipulated colony. We did a field test at Nietver Bay in Stellenbosch, which we wanted to calculate the number of unmanaged honeybees foraging in that area. We did this by excluding all managed colonies for a specified time period, while observers recorded the number of honeybees every 2 minutes for 20 seconds in set areas. The counts were done by 4 people at a single patch, and each person counted the number of honeybees in a smaller area. If we look at the results, in these figures we have the number of honeybees on the y-axis and time on the x-axis, and the highlighted area is when the hives were closed, the managed hives. And as we can see, there's a trend of the number of honeybees actually increasing when the hives were closed. This is not what we expected. If we look at day 2, we closed the hives on 2 occasions, and again there was no significant effect of the hives being closed in terms of the number of foragers, honeybee foragers observed. The close, middle and far refers to that patch's position relative to the manipulated hive. So our results and conclusions: our hive closure method finally worked after some trial and error, and we were able to shut down the colonies with bees accumulating outside when they returned from the field and waiting for the hive to reopen. However, closing 4 hives in our experiment, which represented about 40,000 bees per hive, did not visibly change the honeybee forager counts in the flower patches or transects that we surveyed. This method is thus not quick and easy to work out the pollination requirements of deciduous fruit orchards. We estimate for this method to be usable, one would have to close about 10 times more hives, which is practically not feasible. We would like to acknowledge Hortgrove for funding and sticking with this project, and the 3rd year students of the Department of Botany and Zoology, Stellenbosch University, for assisting with the honeybee forager counts. Below are 2 relevant references. Thank you very much.